9 a.m. In my, let's see, in my watch, 9 a.m. PST. And I hope that you are all healthy and well. And thank you for connecting to another Global Immuno Talk. My name is Elina Zúñiga from the University of California, San Diego. And before I introduce our fabulous speaker today, I would like to remind you that next week, our speaker is Dr. Eike Latz from Germany. So I hope that you can all join us next week as well. And so let me, I see that I have an echo. So let me check this before I continue. Okay, I think that we should make it. Okay, and so now it's time and my pleasure to introduce to you our fabulous speaker today, Dr. Feng Shao. So Feng is a native from China and is based in Beijing. He is an investigator and deputy director at the National Institute of Biological Sciences in Beijing. He was a chemistry undergraduate of Peking University and obtained his PhD from the University of Michigan in 2003. And before joining the National Institute of Biological Sciences in Beijing in 2005, he was also a Damon Runyon postdoctoral fellow at Harvard Medical School. Dr. Zhao's research lies at the interface between bacterial pathogen and host inflammation. A very important topic for sure, and also very importantly, he has made really outstanding advances in this field. For example, Feng identified many of the known cytosolic receptors for bacterial molecules, including caspase 11-4-5 for sensing LPS and alpha kinase 1 for sensing ADP heptose, which is a precursor of LPS biosynthesis. These studies represented very important breakthroughs as they broke the dogma that LPS is only recognized by the membrane toll-like receptor 4 and explain why blocking just toll-like receptor 4 failed to treat sepsis and therefore reopening the door for new anti-sepsis and new anti-colitis drug development. Another impressive example of Feng's research has been the discovery of the gasdermin family of pore-forming proteins and the redefinition of pyroptosis as gasdermin-mediated program necrosis. He identified gasdermin D, whose cleavage by caspase 1, 4, 5, and 11 determines pyroptosis, which is critical for septic shock and other inflammatory diseases. He also showed that gasdermin E is activated by caspase 3, which occurs mostly in non-cancer cells and contributes to cytotoxicity of chemotherapy drugs. And more recently, he demonstrated that pyroptosis is critical for lymphocytes cytotoxicity of the target cells, and that gasdermin activation can stimulate potent anti-tumor immunity. So these were amazing breakthroughs, as I mentioned before, that have been published in top-tier journals and have been recognized by numerous awards that Feng was obtained throughout the years. And as an example, we can mention the Future Science Prize, the Howard Hughes Medical Investigator International Early Career Award, and the Protein Society Irvine Siegel Young Investigator Award, again, among many others. Feng is also a member of the Chinese Academy of Sciences and the German National Academy of Sciences Leopoldina, as well as an associate member of EMBO and a fellow of the American Academy of Microbiology. So I am sure we will learn much more about gasderming and pyroptosis, and we will be delighted by Feng's talk today, which is entitled Pyroptosis in Antibacterial and Anti-Tumor Immunity. So Feng, thank you so much for accepting the invitation to be a global immune speaker. I know it's very late in Beijing, so particularly thankful that you made the effort to connect live with us today.
Thank you.
So, as I mentioned to you, we always like to know our speakers better, and you had an amazing, tremendously successful scientific career, and I'm sure it was very fulfilling with all the impactful discoveries you have made. And so we'd like to ask you a question that may help young and maybe not so young scientists in the audience for their own career. And so the question for you today, Feng, is what would you advise to your 25-year-old self if you were to start your scientific career again?
Thanks, Elena. Thanks for bringing me to the Global Immuno Talk. So at 25 years old, and I just started my PhD study at the University of Michigan. And in retrospect, I think I do have advice. In the end, I had a pretty successful PhD career, published a science paper in less than 4 years. But yeah, I think it was in my third year, I almost quit my PhD study because I was pretty successful during my undergraduate and master study in China. When I went to Michigan, I did my PhD with Jack Dixon. I was a biochemist. So I was not so patient at that time. After 1.5 years, less than 2 years struggling, my project did not move forward at all. I was so worried that I may not be able to defend my thesis and get my PhD. So I started to lose confidence on myself, and I almost start to switch my career to biostatistics. And actually, I did pick some classes course to take. And but I stick for 2 or 3 months, and then my project start to work. So I think if I did quit at that time, I almost quit actually. If I did quit, I would not have a scientific career like what I have now. So and the reason for that, I was not so patient. I think so. For young students, I think the most important thing during your PhD study is be confident on yourself, on your scientific pursuit, and try to learn as much as possible. And indeed, the publication at that stage is not that important. The most important thing is to learn how to do science, how to think about questions, and how to solve the problems and overcome the challenges you might encounter during your PhD study. So and stick on it, and be patient, and be confident on yourself. And that's my advice. Yeah.
That's a terrific advice, Feng. I'm sure super helpful for students in the audience. And you know, even postdocs and faculty, right? At some point you struggle during your scientific career. It's important to be patient and continue. And yeah, thank you so much. I'm sure this is super helpful. So Feng, we cannot wait to hear your talk and learning from you. So if you wish to share your screen.
Okay, I'll start to share my screen. Yeah. Oh, can you see my screen now?
Perfect. Thank you so much.
Yeah. Welcome everyone to today's global immuno talk. And it's pretty late in Beijing now, but I try my best to do a good job. Today, I would like to share our work on pyroptotic cell death with you. And pyroptosis is a quite hot topic at this moment. And I'll take you through some of the historical background and give you an update on the recent development in the field as well in my own lab. So I have to admit that I'm not trained as an immunologist. I was trained as a hardcore biochemist, and you probably will see during the rest of my talk. So my lab started in 2005, and we originally studied bacterial infection, bacterial pathogenesis mechanism. And a few years later, I started to expand my research program to innate immunity. So we want to ask the question, how does the innate immune system deal with bacterial infection? So in innate immunity, we know this toll-like receptor pathway, which is a paradigm of innate immune sensing of microbial infections. So we realized at that time that the toll receptors, which are all on the membranes, and they can recognize extracellular bacterial infection. However, there is another group of bacterial pathogens like Salmonella typhimurium, Shigella fractionarii, even tuberculosis. And those bacteria, they invade into the cytosol, as this right picture shows you. So when those bacteria invade into the cytosol space of the host cells, you could imagine that the toll receptor sitting on the plasma membrane will not be able to recognize intracellular bacterial infection. So we then ask the question for the lab that how does the innate immune system deal with bacterial infection in the cytosol space? Can we discover some novel receptors? Can we identify some new players in this intracellular innate immune sensing or defense pathway for intracellular bacterial pathogen? So here I'm going to show you a very simple experiments using this movie made by at Mel in North Carolina. And in this experiments, we basically simply infect macrophages with intracellular bacteria like Shigella or Salmonella, as I mentioned. And at the same time, we add some propydium iodide into the culture medium. As you know, normally healthy cells will not uptake the DNA dye, the nucleic acid dye, the PI. However, after the cells being infected by intracellular bacteria, you can say that the cells turn into red. The dye, the DNA dye, went into the nucleus and stained the DNA, and you can see that the nucleus is already overstained. And this simple experiments suggest that the infected cells undergoes explosive lytic death, and which we now call pyroptosis. So this not only occur with those Shigella or Salmonella infections, but also many other intracellular or even extracellular bacterial infections. In addition, in immunology, we know there's another group of molecules called the DAMs, endogenous danger molecules such as ATP, and they can also, like ATP, it can also stimulate the macrophages to undergo pyroptosis. So, and this cell death, to understand, there has been two misunderstandings in the history for this kind of cell death. So the cell death was originally observed in 1986 and later on in 1992. But for a long time, it was misregarded as a bacterial valence mechanism. So very intuitively that people think that bacteria want to kill us, right? But indeed, this was not true. This is not a bacterial valence mechanism. The cell death is indeed is a host immune response. The cells sense bacterial infection and then commit suicide to mount inflammatory response to defend against the infection. The second misunderstanding is that because in 1996, Atula Ziklinski's lab showed that this cell death requires a caspase, specifically caspase 1. So for some of the people may know that in the 90s, caspase family is tightly associated with apoptotic cell death. So the caspase family has been deemed as a hallmark for apoptosis. So for a long time, this cell death was also misregarded as a special form of apoptosis. So indeed, caspase 1, which is key for pyroptotic cell death historically. So caspase 1 indeed was the first member of the caspase family discovered, and it was originally purified as an enzyme to process the interleukin-1 beta. In this two papers published in 1992, as a very classical biochemical fractionation study, very beautiful. So those investigators, they want to understand how the pro-I1 interleukin-1 is converted into mature I1 by a proteolytic cleavage, and they identify the proteolytic enzyme. The original name was interleukin converting enzyme, which we now know as caspase 1. So by origin, caspase 1 was discovered in the context of inflammation and cytokine biology. So it also processed interleukin 18, later on other people showed. So basically caspase 1 can induce pyroptosis and also process interleukins. So if we think this cell death is a host immune defense response, so we need some upstream sensors to activate caspase 1 in response to sensing of bacterial infection. And this goes to this so-called canonical inflammasome pathway. So the inflammasome is a cytoplasmic multi-subunit complex. And among the complex, we have receptors such as NRP3, null-like receptors, and they will recognize the infection signal or danger signals in the cytosol, and then recruit adapter protein called ASC, and then further recruit caspase 1 to form the inflammasome complex. So we identified the NIBS, the null-like receptors, as the receptors for flagellin and the type 3 secretion apparatus in gram-negative bacteria. And we also identified, characterized the pyroinflammasome for sensing the bacterial toxin activity. And that was published a long time ago. And quite a few years ago, Richard Dixon and other lab and our lab showed that caspase 11, which is very similar to caspase 1, is most closest homolog, parallel of caspase 1. In caspase 11 in human is 4 and 5. And those caspase, they can directly recognize lipopolysaccharide in the cytosol and then also stimulate pyroptosis, but without the processing of interleukins. So this is so-called a non-canonical inflammatory pathway mediated by LPS activation of caspase 11 is only for pyroptotic cell death, but not for cytokine maturation. As I mentioned, that this cell death was long time regarded as a mixed with apoptosis. So in this movie, I will just illustrate to you what is really the difference between apoptosis and the pyroptosis, right? So on the left side, you will see the cells undergo typical apoptotic cell death. You see that the apoptotic cells, that they shrink the cell body fragmented into small vesicles called apoptotic body. And the key, the critical information here is that for apoptotic cells, the membrane integrity is maintained. So without the disruption of the plasma membrane. So and at the same time, you have phosphatidylserine flipping from the inner leaflet of plasma membrane to the outer leaflet. So that serves as a eat me signal for macrophages to clear the apoptotic cells, the debris of apoptotic cells. So basically, apoptotic cells will not trigger inflammation. It's a clean death. On the right side, you see the cells undergo pyroptosis, and the cells do not shrink, but instead they swell. You see bubbles blowing out from the plasma membrane, and eventually the cells undergo burst with membrane lysis, and then release cytosolic contents and to trigger strong inflammation. right? So and also you release IL-1 beta and IL-18. So we now think is that apoptosis is more a clean death for organism development. It's more a physiological response, while pyroptosis is more like a pathological response, and it is when the organism have a stress, and then you may undergo pyroptosis to send out the signals to the immune system to induce inflammation. So what is the execution mechanism for pyroptosis? And it requires caspase 1 or caspase 11, right? So in 5 years ago, more than 5 years ago, 2 graduate students, Jian and Yue Zhao, in the lab, they took advantage of the newly coming at that time the CRISPR-Cas9 technology, and performed a genome-wide screen to look for genes that are required for caspase 1 and 11 to induce pyroptosis. And both screens hit the same gene called gastromin D. So basically, gastromin D is absolutely required for both caspase 1 and caspase 11 to induce pyroptosis. So gastromin D encodes a protein about close to 500 amino acids. And the 3-dimensional, you can see that it has 2 domains, N-terminal domain colored in blue, C-terminal domain colored in green. And these 2 domains actually bind to each other and they keep the protein stay in an auto-inhibited state. And then between the 2 domains, there's a long linker region. And there's a caspase cleavage site after this FLTD sequence. So you can see, you can imagine that the 2 caspase will cleave the protein in the middle. And then this N-terminal domain, when encounters a membrane, will undergo a dramatic conformational change to form this long 4-stranded beta sheet. At the same time, and this N-terminal domain will oligomerize and form pores, form a giant pores, and get inserted into the membrane. As you can see in this negative stain EM picture, there are multiple pores formed by the N-terminal domain of gastromin D. And this is the cryo-EM structure determined by Hao Wu's lab at Harvard. You can see there's a very giant pore. The inner diameter is about 18 nanometer. For each pore, you have 24, 27, or 28 protomers, this N-terminal domain. And the pore is quite large, as mentioned, and we now know that this pore serves as a conduit for IL-1 beta or IL-18 to be released from the inflammatory cells. So when a plasma membrane have multiple gastromin D pores, and it will disrupt the osmotic potential, and the water will come in, and the cells will swell and eventually undergo burst. So identification of gastromin D and the elucidation of the biochemical mechanism basically give a completed picture and what is really the pyroptosis. So pyroptosis is executed by a pore-forming protein of gastromin D that is activated by cleavage by caspase 1 or 11. So how do caspase 1, 11 become activated to recognize gastromin D? And I'll just very quickly summarize this paper to you published last year. It was mainly done by a graduate student, Kun Wang, in the lab. So we determined the crystal structure of caspase 11 in complex with gastromin D. So here is the C-terminal domain of gastromin D, the inhibitory domain, right? And the caspase have this little, the dimer, caspase is a dimer, form this little beta sheet to recognize the C-terminal domain of gastromin D. There is this very strong hydrophobic interactions. And because of this strong hydrophobic interactions, and basically the caspase can recognize gastromin D specifically and easily. And this will allow and push the cleavage site, this FLTD sequence, get inserted into the catalytic pocket of the caspase. So, and this renders a tetrapeptide independent cleavage. So as you can see in this data, if we mutated this FLTD sequence into triple alanine followed by the aspartic acid, because all the caspase cleave after an aspartic acid, and the dogma says that the 3 amino acids preceding the aspartic acid is critical for the substrate to be recognized by the caspase. However, when we mutate the FLTD into triple alanine and do the in vitro cleavage by caspase 11 or caspase 4, you see that Y-type gastamine D gets cleaved into 2 fragments, and the triple alanine mutation is comparably cleaved. There's little difference between Y-type and the triple alanine mutant of gastamine D. And in the cells, you can see that the triple alanine mutation has no effect. It's still, the cells still undergo cell death with cleavage of gastamine D. So structurally, basically, this exoside-mediated enzyme substrate interaction renders a tetrapeptide independent cleavage of gastamine D. So the same mechanism also applies to caspase 4 and as well as caspase 1, as shown by this co-crystal structure of caspase 4 and caspase 1 in complex of gastamine D. Is very much almost identical enzyme substrate recognition mechanism. So as mentioned, that pyroptosis was originally discovered in bacteria-infected cells, right? Cells undergo pyroptosis in response to bacterial infection. And this is not a bacterial virulence mechanism, but instead is a host immune defense response. So if we made a knockout, gastamine D knockout mice, and infect mice with some gram-negative bacteria like chromobacterial violation, which is a gram-negative pathogen. And this bacteria is recognized by the NAP NRC4 caspase 1 gastamine D inflammasome pathway to reduce pyroptosis. You can see that Y-type mice can deal with, can resist, clean the infection. But if we knock out any gene in this canonical inflammasome pathway, the caspase 1 canonical inflammasome pathway, if we knock out NRC4, if we knock out gastamine D, the mice become susceptible to chromobacterial infection. They died within 1 week. And this is because you have overgrowth of the bacteria in the liver and the spleen of the infected mice. So basically, if you knock out this pathway, gastamine D or caspase 1, the mice cannot clear the infection, and the bacteria, they just replicate heavily in the mice. And this is also true for the non-canonical inflammasome pathway using Burkholderia terradensis as a model. You can see that the Y-type mice can clear Burkholderia infection, but the caspase 11 knockout mice and succumb to the infection. And the Burkholderia is an intracellular bacteria. It's a gram-negative, will leak out LPS to be recognized by caspase 11 and gastamine D. And there's a similar bacteria called Shigella, which is, we have a long time interest into this organism. Shigella flexneri is also intracellular free-living bacteria, which will inevitably expose this LPS to the cytosol, to LPS, to caspase 11. However, the Y-type caspase 11 mice showed no difference in response to Shigella infection. Both of them succumb to Shigella infection, and we were quite intrigued by this. And to cut the long story short, we found that Shigella has a virulence factor, a type 3 factor called ASP-C3, and this factor can, using a sugar donor, a sugar ligand, to post-translational modify caspase 11 and caspase 4, and inactivate this caspase to block gastamine D activation. So if we delete OSPC3, ASP-C3 from Shigella, in this green line, you can see that when you infect the mice with the mutant bacteria, now the mice can survive very well and can clean the infection, and the bacterial load, the bacterial load of the mutant ASP-C3 mutant bacteria is very low compared with that in the caspase 11 knockout mice. So, and this study highlights the importance of the LPS caspase 11 pathway for defending against the intracellular gram-negative bacterial infection, because that's why Shigella has evolved a specific virulence factor to shut down the caspase 11 pathway. And now using this OSP-C3 mutation, the mutant bacteria, we found that if we can see that the mutation can cause increase the antibody production, anti-Shigella antibody production. Because if you delete OSP-C3 from Shigella, now the host can activate caspase 11, induce pyroptosis in the mice. And after activation of this LPS caspase 11 gastromin D pyroptosis pathway, the mice can have a higher level of anti-Shigella antibody. And if you do the same experiments in caspase 11 or gastromin D knockout mice, you don't see the increase the antibody production in response to the OSP-C3 deletion mutant infection. And if we re-challenge the mice after the OSP-C3 infection, and the mice are better protected from re-challenge by wild-type bacteria. So, and this also depends on caspase 11 or gastromin D. So basically, this study suggests that if we immunize mice with the attenuated OSP-C3 deletion Shigella mutant, and this will give us a vaccine-like protection in a caspase 11 gastromin D dependent manner. So this study shows that this non-canonical LPS caspase 11 pyroptosis pathway can promote anti-Shigella antibody production, which can protect mice from Shigella reinfection. So basically, these two infection studies highlights the importance of pyroptosis, the protective function in the immune protective function of pyroptosis, right? However, we know that the immune response is always a double-edged sword, right? If the pyroptotic program goes excessively or is out of control, they will bring a deleterious effect. Then for a typical example is that if we inject mice with very high dose of lipopolysaccharide, it could be as high as 50 milligram per kilogram. And this will induce endotoxic shock, which mimics the septic shock in human. And you can see that wild-type mice die within 1 day in response to this high dose endotoxin injection. But the caspase 11 and gastromin D knockout mice, they can stay alive very well. So this data suggests that systemic pyroptosis is likely the key for septic shock. And now we think that if you block this pathway, either inhibiting caspase 11 or inhibiting gastromin D, you may have a chance to develop antisepsis, new antisepsis drug. And this has now been pursued in the biotech industry. What's more interesting is that we not only have gastromin D indeed belongs to a larger gastromin family. In human, there are 5 members, gastromin A, B, C, D, E. And the gastromin A has 3 genes in mice, A1 to A3. Gastromin C has 4 genes, expanded into 4 genes in mice, 4C1 to C4. And this family is highly conserved in lower organism, and even coral and hydro. And now there are also papers showing that this family, this gastromin-like protein can also be identified even in bacteria and the fungi. So all the family members, they share this 2-domain architecture, N-terminal domain that can make pores that is normally inhibited by the C-terminal domain. But the difference is that other gastromin family members, they cannot be cleaved by caspase 1, caspase 11, but they can be activated by any other protease cleavage. So as long as you can cleave any place within this linker region, or you have another way to break up the 2-domain, to break up the auto inhibition, you can activate the pore forming domain. So we think that for pyroptosis, the key, the hallmark is not caspase, it's gastromin family of pore forming protein. That is the hallmark of pyroptotic cell death. So now I'm going to summarize the story of gastromin E, another gastromin family members. So now getting back to the apoptosis studies, we know that caspase 3 can activate apoptosis in response to DNA damage or in response to caspase 8 or TNF receptor activation. So, and this is true that if we damage the DNA, by treating cells with chemotherapy drugs such as cisplatin or doxorubicin, and the cells will undergo apoptosis, and you can see this very beautiful apoptotic morphology in HeLa cells, and this is due to activation of caspase-3. However, when graduate student Yupeng express gastamine E in HeLa cells, generate stable cell line, we do the same kind of stimulation. And we did not see apoptosis, but instead the cells develop those characteristic bubbles and undergo burst, eventually die out of pyroptosis. And this experiment suggests that an ectopic expression of gastamine E in HeLa cells will switch caspase-3 mediated apoptosis to gastamine E mediated pyroptosis. And why is that? And this is because gastamine E has the caspase-3 cleavage site in the interdomain linker region. And indeed, in vitro, we can show that caspase-3 can cleave, but not other caspase. Caspase-3 can efficiently cleave gastamine E in vitro into two domains, N-terminal pore-forming domain, C-terminal inhibitory domain. If we mutate the aspartic acid residue into alanine, we can block the cleavage, right, as expected. So now we can put the mutation into the HeLa cells to block a caspase-3 cleavage. So you can compare that. This is the previous experiments that gastamine E expression switch into pyroptosis. But if we make a mutation, the cells go back to apoptosis. So this data suggests that it is caspase-3 cleavage of gastamine D that drive the cells into pyroptosis rather than apoptosis in the native HeLa cells. So, and this is because pyroptosis has much faster kinetics than apoptosis. So if we have caspase-3 activation, you go into apoptosis in the absence of gastamine E. However, in the presence of gastamine E, which will be quickly cleaved by caspase-3, and then you will switch the cell death to a pyroptosis because of the faster kinetics. So, and if in fact, gastamine E is not expressed in many cancer cell lines. In this NCI 60, we have profiled probably 100 cancer cell lines. Only about less than 10% of cancer cell lines have gastamine E expression. You can see most of them do not express gastamine E. And this is because epigenetic silencing. So during cancer progression, tumorigenesis, the promoter of gastamine E, is methylated, is epigenetically modified, and then the gene gets silenced. But in some normal cells, non-cancer cells, and this shows 5 primary human cells. You can see that 3 of them highlight in red, such as epidermal keratinocytes, placental epithelial cells. They express endogenously gastamine E. This umbilical vein endocellular cells, they do not express gastamine. So for the negative gastamine E negative cells, if we treat with DNA damaging drugs such as doxorubicin, they undergo apoptosis, just as the dogma or textbook tells you. However, in this gastamine E positive cells, you treat with doxorubicin or other chemotherapy drugs, you see the big bubbles coming out. They show very characteristic pyroptotic morphology. So this data suggests that in our body, we have a normal non-cancer cells. Some of them express endogenous gastamine E, and those cells will develop a pyroptosis in response to DNA damage, in response to caspase-3 activation. This data also suggests that those DNA damaging chemotherapy drugs, or even maybe radiation therapy used in cancer therapy in clinics, and those agents treatment, they induce cell death in vivo, not only involves apoptosis, which is a traditional view, but may also induce pyroptosis in vivo in cancer patients. And that those cells, they are different in vivo from we grow them in the cultural system. So This slide just diagram just summarizes to you that in cancer cells, they have epigenetic silencing of gastromin E expression, and they undergo apoptosis in response to DNA damage by chemotherapy drugs. However, we have normal cells or some outlier cancer cells. They have gastromin E expression, and in response to DNA damage by those chemo drugs, they develop a pyroptosis, which is highly pro-inflammatory and cause tissue damage, inflammatory damage. So this pathway I think we have two functions, and it probably is contribute to the toxicity of those chemotherapy drugs, right? And it also may stimulate inflammation, immune response, and stimulate antitumor immunity, which I will show you later on. So we have two gastamins, gastamin D from the inflammasome pathway, which is activated by caspase 1 and 11, cleavage in the middle, and then release the pore-forming domain to induce pyroptosis. Gastamin E is activated by caspase 3, which probably functions in many biological pathological contexts. And the consequence is the same. It release the pore-forming domain and induce mediated pyroptosis. So the concept I want to share with you is that, so we usually tackle caspase, we use a tag caspase with some kind of biological function, which is not true. Caspase is simply a biochemical activity, just like a kinase. And think about it a little bit. So it's just a simple biochemical activity. It does not equal apoptosis. And it's not the caspase, but it's the substrate that eventually determines the nature of cell death, right? So if the caspase cleave a pore-forming protein, such as gastamin D or gastamin E, and the cells will undergo pyroptosis, right? That's, I think, is a very important concept. We also found that many gastamins, particularly gastamin E and gastamin A and C, it's very hard to find their expression in cancer cells. They seem to be silenced, tend to be silenced in cancer cells. But why is that? So in this collaboration work with Dr. Zhibo Liu at the Peking University, and he's a chemical biologist, and he developed a very sophisticated chemical linker. And the linker can be cleaved by a small molecule of phenylalanine mimetic called trifluoride of phenylalanine. So you can just imagine that this chemical linker is just like a rope. We can use this rope to lock gastamin, to link gastamin to nanoparticle. Okay, so nanoparticle will be engulfed or endocytosed by mammalian cells and particular cancer cells, right? And this gastamin is conjugated to nanoparticle and goes inside cytosol through the endocytosis pathway. And then we can have this small molecule, which just like a phenylalanine can be imported into cytosol through amino acid transporter. So the nice thing, and this small molecule will cleave the linker, just unlock it, right? And then gastamin can be released and to make pores in the plasma membrane and induce pyroptosis. The nice thing here is that this nanoparticle conjugate and the small molecule, when you inject them into mice, they converge onto the tumor site. So that's why we call it as a bi-orthogonal reaction for tumor selective protein activation, right? So if you inject the nanoparticle conjugate gastamin into mice, and then you inject a mice with a small molecule, and the reaction, the cleavage reaction will only occur on the tumor site. So we did this syngeneic tumor model, this 4T1 triple negative breast cancer tumor model in the mice, and then at the day 6 we inject the mice with nanoparticle, and then followed by the small molecule to release the gastamin to induce pyroptosis. Then we did 3 rounds of induction of pyroptosis, and after 3 rounds of induction of pyroptosis, you can see that we see a very dramatic tumor shrinkage tumor regression phenotype, and statistics shows very striking. And we have all the other controls. We can even make a point mutation to knock out the pore-forming activity, and we don't see the tumor shrinkage phenotype. And then either in the intratumor injection or intravenous injection, we have the same effect. We also see the same effect in other tumor models, such as the EMT6, another breast cancer tumor model. So you can still imagine that, you probably think that the tumor shrinkage is caused by the tumor cell pyroptosis per se, right? However, this is only partially true, or is not true. So this method, this chemical biology method that allow us to activate it, release gastromin specifically in the tumors in the mice. This method works highly efficiently in the test tube, this chemical biology reaction. But in the cell culture, we can only induce around 30% or 20% of cell death. So you can imagine that the cells has to endocytose the nanoparticle, and the same cells has to uptake the small molecule, right? And then the cleavage reaction can have to occur in the cytosol to release sufficient amount of gastromin to kill the cells. So you can see that, you can imagine that the efficiency will be very high. So in vivo, we can only kill about 10% of tumor cells in the mice. But if we only kill 10% of tumor cells, where is the rest of 90% of the tumor cells? Why they did not grow into tumors, right? So then we did experiments in the athymic T cell deficient mice. So in the wild type BRCA mice, the previous experiments, you see when we induce pyroptosis, the tumor did not grow. We see a dramatic tumor shrinkage phenotype. The same experiments when we did it in the athymic T cell deficient mice, there was absolutely no tumor shrinkage phenotype. So this data suggests that the tumor shrinkage caused by pyroptosis, a few cell pyroptosis, cancer cell pyroptosis, requires the T cells. So basically, we think that a few tumor cells undergo pyroptosis, and pyroptosis triggers the inflammation. It's part of the immune defense response. It would trigger inflammation and remodulate the tumor microenvironment, and then that will activate the T cell media and the tumor immunity. So even if when we reduce the 3 rounds of pyroptosis induction in this 4T1 tumor model into 1 round of pyroptosis, is not efficient. The inflammation is not efficient to induce sufficient antitumor immunity. So we know that this 4T1 tumor model is a cold tumor. It cannot be, is not sensitive to the checkpoint blockade of the anti-PD-1 treatment. So you see, we treat mice with anti-PD-1 antibody. There's no tumor shrinkage phenotype. However, after 1 round of induction of pyroptosis, we then are followed by anti-PD-1 treatment. We now see a dramatic tumor shrinkage phenotype. So this data suggests that a little bit of tumor cell pyroptosis, although by itself is not sufficient to trigger a strong, robust anti-tumor immune response, but it can convert this so-called cold tumor into whole tumor, so that the tumor can now be sensitive to the checkpoint blockade therapy. So this data suggests that the gastromin activation induced pyroptosis can trigger a very potent anti-tumor immunity. This explains that why the tumor cells tend to silence gastromin expression, because probably during tumorigenesis, there's a reprogramming, epigenetic reprogramming, and they want to, the tumors want to silence those pore-forming proteins, gastramins. And this probably explains very well that in the clinics that, now there are a lot of combination trials using checkpoint blockade with chemotherapy or even radiation therapy that seems to have a very good clinical efficacy, because we show that the DNA damage induced by chemotherapy and or radiation therapy can induce gastromin E activation, and gastromin E activation basically will give the same kind of a pyroptotic response. And lastly, I want to share another piece of work that in this work, we want to understand that we asked the question, does gastromin-mediated pyroptosis normally play a role in antitumor immunity? So why we want to ask this? So we know that in antitumor immunity, downstream of TCR recognition of the MHC presented tumor antigen, when you have an PD-1 to release the checkpoint, right? And then that will activate the cytotoxic T cells. The cytotoxic T cells will then use granzymes to kill the target tumor cells. So granzymes are sort of the execution protein secreted by the CTLs and delivered into, through periphery mediated, delivered into the target cells, into tumor cells. And then granzymes are serine protease. There are 5 granzymes in human, 11 in mice. And historically, granzymes mediated killing was thought to be apoptosis, because in the 90s, the immunologist was highly influenced by cell death people, that the cell death people think that granzymes are pretty much alike or similar to caspase, and the caspase induce apoptosis, and the granzymes probably also induce a similar cell death like apoptosis. Indeed, granzyme B, the best studied, the most extensively studied granzymes can activate a cleave caspase 3. So, and there's the textbook, immunology textbook tells you that granzymes will induce apoptosis and kill target cells, including tumor cells, through apoptosis. Is this really true? So in these experiments, I'll just show you that we use NK cells as a model. We can you also use the CTLs. The NK cells does not require TCR recognition, so it's relatively simple. So in 293 T cell, we use NK cells to kill 293 T cells in an in vitro co-culture system. So we load a fluorescence dye into 293 T cells, and the dye gets in, then cannot get out. And it's a green dye unless the cells undergo lysis. So in the left side, you see the small cells are NK cells, and they try to attack the 293 T cells, but we did not see the green fluorescence leakage from 293 T cells. So there's no lytic death, right? However, when the postdoc fellow Zhiwei Zhou in the lab, he expressed the gastromin B, the third gastromin I talked about today, into 293 T cells. We did the same kind of killing, co-culture killing assay. We also added PI, propylene iodide, into a culture medium. Now you can see that when NK cells attack this gastromin B expressing 293 T cells, the green fluorescence disappears and the PI gets into the cells. So this suggests that gastromin B expression, but not any other gastramins, expressing in 293 T cells can also confer pyroptotic killing of 293 T cells by NK cells, as well as CTLs, which I did not show you. So why is that? So we have 5 granzymes, and to cut long story short, we found that specifically granzyme A can efficiently cleave gastromin B into 2 fragments, the N-terminal domain and the C-terminal domain. So the cleavage also occurs at the interdomain linker region to release the N-terminal pore-forming domain. So very much like a caspase cleavage of gastromin D or gastromin E. So in the granzymes, specifically granzyme A made by CTLs or NK cells, and it can efficiently cleave and activate gastromin B. And this will release the pore-forming domain and make pores, and the target cells will undergo pyroptosis. And we found that endogenously, there are 3, here I'm showing you 3 cell lines, and they are digestive tract cancer cell lines. And they have high level of endogenous gastromin B expression. If you give those cells granzyme A, all the cells will undergo dramatic pyroptotic cell death. And if you make a mutation in this cleavage site, the lysine residues, you can block the pyroptosis, which I don't have time to show you. And so we also tested with cytotoxic T cells using the CAR T cells, right? We know CAR T cells has been approved for cancer immunotherapy, particularly the CD19 CAR T cells. So we use the CD19 CAR T cells to kill 293 T cells that have gastromin B expression. You see that the cells develop those big bubbles, right? You see the leakage of green fluorescence from the 293 T cells. And if you knock down siRNA, knock down granzyme A, you can block diminish the pyroptosis killing. And if you mutate the cleavage site, and there is no cleavage of gastromin B, then you don't see the pyroptosis. And we show the same kind of pyroptotic killing by TCR T cells. You see that in this melanoma cells, A375 melanoma cancer cells, and in response to TCR T cells killing, gastromin B gets cleaved, and the cells undergo pyroptosis, lytic death. If you mutate the lysine residue, you don't get cleavage, you block the TCR-T killing, pyroptotic killing of melanoma cells. So gastromin B, where does it express? And we found that it's highly expressed in the entire digestive tract, starting from the lung to even to the appendix. It is also expressed in the lung epithelial cells as well as the bladder epithelial cells. And it's very interesting. We also found that gastromin B is transcriptionally induced by interferon gamma and to some extent the TNF alpha. For example, in HT29 cells, you don't see gastromin B expression, but if you treat it with interferon gamma or in TNF alpha, you see a dramatic transcriptional induction. And the same applies to many cancer cell lines. So gastromin B actually is a transcription target of interferon gamma. So we know that the cancer and the tumor immunity is driven by interferon gamma. So T cells secrete interferon gamma, and interferon gamma acts on tumor cells. It can induce PD-L1 expression. It can also induce gastromin B expression. So when gastromin B is expressed, and the granzyme A coming from cytotoxic T cells will specifically cleave the linker region in gastromin B and release the gastromin B and make pores, and the target tumor cells will undergo pyroptosis. So the most important thing is not only the death of tumor cells, and this death is different from apoptosis. It will trigger inflammation. It will remodulate the tumor immune microenvironment and to further amplify the immune response to further stimulate antitumor immunity. So in this CT26 colon cancer tumor syngeneic model, we also see gastromin B mediated pyroptosis. It has a synergistic effect with anti-PD-1 checkpoint blockade treatment. So this data suggests that gastromin B as well as gastromin E, and the work from Judy Lieberman's paper show very similar story that gastromin E in response to granzyme B activation also plays a role in anti-tumor immunity. So we think those gastamins may serve as biomarkers for cancer immunotherapy. If cancer cells express those pyroptotic factors, they may have a better chance to respond to immunotherapy. And if we can develop small molecules to activate the gastamin, it could be a next generation or cancer immunotherapy reagents. So I want to acknowledge the students and postdoc fellows in the lab who did all the work. Jian Junsi and Mie Zhao, they originally identified the gastamin D, and Yupeng Wang, Wenqin, and Xu, and they did work on gastamin E. Jin Jingding is a postdoc fellow, showed the pore-forming activity, and together with Kong Wang and postdoc fellow Qi Sun did the structural work. And the last piece of work on gastamin B was done by postdoc fellow Zhiwei Zhou and graduate student Hua Bing. And Qinyang Wang from Dr. Zhibo Liu's lab did a collaboration work on the chemical biology delivery of activation of gastamin. And Feng Shao Wang made knockout mice for us. And I'll stop here. Thanks for listening.
Thank you so much, Feng. This is I feel humbled and inspired by the tremendous work you have done in this field. Really, each of the discoveries you described today are so impressive on their own. So I'm sure the rest of the attendees will feel the same way. And now I would like to guide them to take the opportunity to interact with you in Twitter and ask questions that can be done in an asynchronous manner. And so if you are watching this talk in YouTube, you can ask questions in Twitter after you watch the talk later. And it's late in Beijing, so probably Feng will be answering questions tomorrow or
tomorrow. Yeah, I'll do that tomorrow.
Sounds great. Thank you. And so to ask questions, search for the account Global Immunotalks. Find a tweet that says, ask questions for Dr. Feng Shao here, and reply to that tweet with your question and mentioning the hashtag Global Immuno. And then Feng will use the account Immunospeaker to answer to the questions. And with that, we will close the session for everyone, and I hope that you can join us again next week. So Feng, again, thank you so much for presenting and being a Global Immuno speaker today. Thank you so much.
Thank you. Yeah.
Bye.
Bye.
Bye, everyone. Thank you.
Yeah. Have a good day.